My name's Tom McClelland. As Molly said, I'm one of the fifth-year medical students here at Oxford. And today, basically, what I'm going to take you through is kind of biology, but in the way that we learn about it at medical school. So at medical school, you'll start by maybe looking at something intracellular, looking at the biochemical pathways. And then maybe moving on from that, you'll look at how those cells then interact with each other to form tissues, and then how those tissues form organs. And then overall, you'll start looking at how those tissues interact in health, and then importantly for medicine, how they go wrong in disease. And by doing that, and by starting at first principles and knowing the kind of biochemical pathways, you can end up at the end of the day coming up with medicines that help you treat the diseases that you have started to work on. So today we're going to look, and today's theme really is looking at cancer. So I'm sure you've all heard of the cell division process, have we? Yeah? Lots of happy faces I can see thinking about cell division. I'm sure you've seen a system just like this before, which is the cell division cycle. Now this is a pretty cool bit of biology, to be honest, because we're all sitting here, and believe it or not, you're made up of like millions and billions of cells, but you all started from one cell. So this process is pretty much what you've all got to be thankful for. And as this goes round, you can see how, you see the little red bits, which are checkpoints. And basically, those are the checkpoints that are built into your DNA, built into this process, so that your cells don't divide uncontrollably. You know, this is a really important process. This is how we came to be. So it needs to be controlled, and it needs to have a regulatory system. So as you go through G1, the cell's just growing, and then there's a checkpoint. Let's just check here, are we definitely going to go through with this? Then we go through to the replication stage. So what actually are the checkpoints, all right? So the checkpoints are proteins within the cell. And they're really important in that they're basically sensors. They work to check that everything's okay, and if the conditions are just right, and the cell's happy to move on and to divide, then they'll give the thumbs up, you know, right, let's go ahead, we're happy to go. And to think about this, we need to think of two proteins. One are the CDKs, and one are the cyclins. Okay, so the CDK stands for cyclin-dependent protein kinase, right? Cyclin-dependent gives it a clue. These two are best friends in this process. They need each other to continue on. So what is a CDK? This is essentially just an enzyme, a protein within the cell. And when it binds with cyclin, that's basically the green light. All right, we can move on to the next step. So if the conditions are just right, we can move on. So what's a cyclin? So cyclins are lots of little molecules that are highly influenced by the environment that they're in. Essentially, you've got these molecules that are just waiting for the right temperature, the right kind of process within the cell, and then they'll all combine and allow the cell to continue to divide. So as I said, basically within medicine, we're going to look at how they work in health, and then how they work in disease. And if I'm going too quickly, let me know. If you've got any questions, just stick your hand up. So that's it in health, right? What's it look like in disease? So cancer is essentially a process by which cells in the body have lost that kind of regulatory pathway. You know, the cyclins and the CDKs are not working the way they should be, such that the conditions might not be perfect, and the body might not even need the cells to divide. But because the processes have been lost, and there's been mutations in genes that produce the proteins that kind of control this whole process, there's been a real breakdown in that communication link. So, as we said, the checkpoints are vital, really, aren't they? Like, if you're going to go through with quite a big process of dividing in two, you want to make sure that you're doing it right. So, essentially what happens in cancer, you've got too many green light signals. You've got uncontrolled cell division, too many yeses and not enough, whoa, slow down here, we don't need to have any more cell division. And I'm sure you've seen in news and about smoking cigarettes, UV light, and lots of other carcinogens, even cooking your toast too long in the toaster, which I saw in the news recently. These things can lead to, you know, kind of these mutations, but please don't worry about the toast you had this morning. So essentially, this is what we're looking at here. This is how we would look at it in a lab. You've got normal cells that are responding to growth factors, signals to say, yep, go ahead. And on the other side, you've got cancer cells. And whether there's growth factors or not, they're just going ahead. You know, the brakes are off. So this is basically just a diagram of how these growth factors work. They bind on the outside of the cell, because the cells need to know what environment they're in. So if they're in the right environment, then they can do this pathway, as you see, going down towards cell division, which is basically taking the brakes off the cell division process. So in cancers, which I'm sure you know, they form tumours, which are just big balls of cells. They are multi-layered, uninhibited cells that are just rapidly, constantly dividing. And as I said, there are brakes and there are accelerators in this process. We have hundreds of genes that act as, green light signals, and just as much we've got ones that act as red lights to say, whoa, I'm not happy with this, we need to put the brakes on this and maybe not divide. I don't think the conditions are right. So why is this important? Right? So as I said, we've come from knowing the cell process. Okay, we know kind of why it happens and where the checkpoints are. And then we've got, in clinical practice, we see people that have got cancers that are causing big problems within their bodies, and we think, well, how do we put these two together? Where has the mistake happened in this line? And this is when research and working on these things gives you the answers. Has anyone heard of Herceptin? Yeah, some hands up. Good work. So Herceptin is basically a medication that acts just like antibodies in your body does. When you get the cold, or if you get an infected elbow or something, these are the antibodies that fly around and kind of try and attack that process. And what we've done is we've used that, our own body mechanism, and we've engineered special little molecules that look just like those antibodies, and we can give them to people. And what this does, as you can see here, it blocks what's that called, the epidermal growth factor receptor, right? A growth factor receptor, which most likely has gone wrong within the tumour cell. Because if you think about it, if the epidermal growth factor receptor is constantly sending signals for the cell to grow and divide, that's how you're going to end up with a cancer. So we've made a little targeted molecule. When I say we, I didn't do this, I wish I did. And it connects to the cell and basically says, whoa, let's put the brakes on. Let's turn off that green light signal. Let's stop these cells dividing, and therefore, let's stop the cells causing a cancer. So basically, we've gone through first principles, clinical practice, and then how we interact with that on a pharmacological level. And for you guys, what's important here, I think, is when you're thinking about these concepts, it's really, get something you're passionate about, if it's be cancer, kind of know the biochemical process, maybe know about a drug that can interact with that, and then you've got to be in touch with the news as well. These things are constantly on the news. You can't go one day without seeing these in newspapers and online. And bringing that it kind of brings it to real life, because it's easy to think about all these kind of concepts and drugs that we can use, but we have to think about it in the context of the NHS and whether we can afford these kind of things, and that opens a whole new kettle of fish, box of worms, whatever you want to call it. So essentially, that's a very rapid kind of, I don't know, kind of what happens in cancer, and I hope that's given you a little taster of what we learn in medical school. So the question was, where does all this fit in within your medical training? Which is a fantastic question. So where I started at the start with the biochemical processes, that might be in first year, learning about the intracellular things, progressing through to maybe in second year learning about the pharmacology of everything and how the Herceptin works and how antibodies would work, and maybe progressing through into clinical school where you'll actually see people who unfortunately have cancers, and then you deal with them and you speak with them and kind of ask them questions and do more diagnostic things, and then move on to treatment when you become a doctor.
Earlier about how certain cells, like cancer cells, do not need growth factors because they sort of are out of control. And I think that's a really important question, and this is what is so interesting about cancer, which is why it's something I'm very interested in reading extra things about. But essentially, if you can imagine, you've got a cell, it has loads of different receptors on it for different growth factors. One of them, in this case, is called HER2, and it's actually it leads to downstream reactions, which essentially means that, so you can imagine with antibodies, do you remember lock and key, whenever you did that in GCSE? So essentially, the receptor is sitting on the cell, and the growth factor will go in and make a sort of lock and key, so that it gets like an activation. That activates a series of reactions, and that's what's called a downstream pathway, because sometimes I find that really confusing. So in this case, if you want to read up more about it after this talk, you can look up the HER2, and you might want to know that the name of Herceptin, the general name is called trastuzumab, TRASTUZUMAB. So all of these drugs will have a generic name that has mab at the end for monoclonal antibody, so that's quite nice to know. And then they have their brand names, which is how they sort of promote them and try and get them into clinical practice. But what your question about the growth factors is that, yes, cancer's out of control, but certain growth factors can be at the root of what's causing the extra growth. So in other cancers, it can be, for example, estrogen receptor, which is a hormone. So the hormone can be causing the cancer to grow out of control. And in some breast cancers, it's this epidermal growth factor that's out of control. So whilst you're not going to solve the whole thing with just these monoclonal antibodies sometimes, if you can find the defect in the cell that's causing it, so if you can find that it's the HER2 receptors that are really overexpressed on the cell, because sometimes what happens is, if you can imagine you've got a round cell, like that blue circle that you can see in the presentation, imagine all around it you've got little receptors for growth factors. Now in a normal cell you've got lots of different ones, but in some defunct cells you can have a disproportionate number of a specific growth factor, like HER2 in this example. So if we know that the breast cancer is affected by that, then we can block some of those and reduce the drive that's causing it to grow. It is quite complicated to get your head around, and so you definitely will need to go away and do some reading on the bits of this that you find interesting. But it's about trying to work out, this cancer cell is essentially, it's growing too much because of overexpression of the receptors or faults in the signal pathways. You can't always say it's just one thing, but the more things we can find specific to the cancer itself, the better our outlook.